Okay.
Thank you. Yeah.
Yeah. It's funny, I cannot see that it's getting recorded, but that's okay. As long as you can see it, that's fine. All right. So I have started FIJI over here, and what it's showing. Oh, actually, what will happen is if I record like this, there won't be any audio.
Okay. So can I? One second.
So let me do this. Let me call the number to join this meeting.
Okay.
In that way, everything that is being spoken
will be recorded as well.
Will be recorded, okay?
Yeah, no problem. Yeah.
I'll resume recording. All right. So, as I said, as soon as you open FIJI, it will always kind of by default take you over here. When you are doing it the first time, I would say, yes, update everything. So everything that is present will be the most up-to-date that you can have.
Okay.
Okay. So I'll do over here saying yes, please, and you will know what will happen. So it will give this warning that the Java is too old to handle as a security risk. It's up to you if you want to update Java or not. But you can say okay, and it will start updating everything. So it's giving me a bunch of errors over here, which you can ignore. That's totally fine. And it's updating all the jar files. So think of, and then it says your image is up to date. Okay, and so okay. Now think of FIJI to be something like the coffee maker in which you end up putting those pods, and you press a button and you get a coffee, right? So depending on the type of the pod, you can have a different flavor coffee.
Yes, yes.
So in the same way with FIJI, you have a base FIJI which is image J2. On top of that, you can have different plugins, which is essentially like the pods of a coffee maker, and depending on the plugin you use, you can do different tasks. Okay. Now there is something called update site. So you can go to manage update site, and it will give you options of lots of different plugins that you can install in FIJI. Okay. Now there are different ways of installing plugins in FIJI. If somebody tells you that, oh, you know what, I want to detect filaments, so you can get this filament detector. So these are the people who are developing these plugins. And as and when they are updating something, when you click on this, FIJI will update everything if they updated something for you. Okay. And sometimes, let's say, for example, if I want to give you an example of it could be anything, doesn't matter what it is. Okay. If I want to download a plugin, sometimes some groups, they have a lot of different plugins. So let's say this is Leiden University, right? And this group over here, their group uses, let's say, 5 or 6 different plugins all the time. They will have that collection. An example of that can also be this plugin which I can install, BioVoxel, over here, right? So let's install BioVoxel. So before I do that,
so Gaurav, just a quick question. So so do we have to do it every time? So suppose like you're showing the filament detector. So every time before doing, do you have to check for?
No.
Okay.
Not every time. You can ignore this. So
Okay.
I'm going through that right now with you. Okay.
Mm-hmm.
So for example, if I go to plugins, these are the basic plugins that we have.
Mm-hmm.
Uh, I mean, some of them are already installed because this is clicked and they're installed. So for example, let's say if I want to install BioVoxel, right?
Mm-hmm.
So that's right over here. And when you go to plugins, you don't see it, right? You don't see any BioVoxel over here. So I'll go and click BioVoxel. I'll click update URL,
Mm-hmm.
say close. I'll do apply changes. So over here it said that once I apply changes, all these plugins will be installed. Okay.
Mm-hmm.
So I do apply changes and it installs everything. Now, before you can execute something, you have to quit FIJI
And reopen it.
and restart it. So now once I restarted, if I go to plugins, now I can see BioVoxel, right?
Okay.
And in BioVoxel, they have all these different plugins which now appear.
Got it. Got it. Yeah.
Okay. So that's nutshell of installing plugins.
Mm-hmm.
Now, if I ever send you one plugin, okay, how do you do that? If it's not part of that repository that I just showed to you.
Mm-hmm.
So if I was to send you a plugin, which is not part of the repository, you can go to plugins and install plugin.
Oh, so you are saying you would be sending a like file, like plugin file, right?
Right. So if I send you like a .jar file,
Mm-hmm.
right, you can go into your downloads folder and select that and say open, and that gets installed in a particular place.
Got it.
Okay.
Okay.
So there are different ways of doing it.
Mm-hmm.
Okay. So that's in nutshell that. Once you come to FIJI, this is the window that you will see. Now, depending on what you have, it will be different because this menu bar is completely customizable. You can customize this. Okay.
Okay. Mm-hmm.
So the first thing you will see is file. You
Mm-hmm.
can open something, but there are other ways of opening. So let's say, for example, I'm gonna pause this because I want to open our files. So
Mm-hmm.
give me a second. I'll pause the recording first. All right. So if I want to open one of the files that we were talking about earlier,
Mm-hmm.
I can just go to my ND2 file and drag it. Okay. Now that will open something
But
called
I, I have
bio.
I have a question here. So just a question I had it other day. So you remember we talked about different depth of the cells, and to get the overall idea of the cells, we have to take the images from different depth, right? Depending on the cell depth, like cell depth. So whatever ND2 file you are generating, is it a single like image of one certain depth of the cell, or is it the stack? Is it all the
So
stack?
ND2 file or CZI file or call it any file. So these are proprietary files for different microscope companies, right? So
Mhm.
ND2 is an icon specific file.
Okay.
Now, ND2 file can be as big as 10 gigabytes. It
Okay.
can store not just one single plane, it can store X, Y, Z as well as time points. So if I want to do X, Y, Z at different time points,
Mhm.
it can also store that.
Okay.
Does it make sense?
Yeah, it makes sense. Got it.
Okay. So it's not just one single stack. It is exactly what you are saving. It's giving you everything.
Got it. Yeah.
Okay.
Mhm.
All right. So the thing with FIJI is, as soon as you drag your file over here,
Mhm.
it gives you this bioformats window.
Mhm.
What bioformats does is it can identify any different file or different types of file from any microscope. So that makes FIJI system agnostic. It does not depend whether you have a Nikon file or a Zeiss file or a Leica file. It can open any file. And FIJI can open any file because of the bioformats viewer. Okay. Now you can play with this if you want to look at the metadata, which is what kind of microscope you used or what kind of objectives you have used and so on. You can enable this. If your file is too big, let's say gigabytes in size, you can use virtual stack so that it does not use a lot of memory. But at the moment, I know that my files are very small, so it doesn't matter. So I can just go and open the way I want to. If I want to split individual channels, I can enable this. If I want to open individual Z-planes in different windows, I can do this. I can enable split focal planes and so on. Okay, so you can play with these options later on. So I'll go OK, and it will open the file the way it is, right? So it opens up the file where you can see different channels and different Z stacks.
Different Z planes. Yeah, now it makes sense what you answered me first here about the ND2 file.
All right. So few things that you should know about FIJI is that FIJI runs on your RAM memory. So depending on the RAM that you have on your computer, you can only handle those big data sets. So if your RAM is 4 gigabytes, you cannot open data sets greater than 4 gigabytes. Now, you have to tell FIJI how much RAM it can use. So this is important for you to set. So you go to Edit, Options, and Memory and Threads. Okay. So I mean this one is set to 50 gigabytes because it's a workstation and it's really super duper awesome. But depending on the RAM that you have, you can set that to about almost the max. Okay. So you are limited in terms of opening a data set depending on the RAM of your computer. So that's something that you have to figure out. All right. So coming back to the data set, it will really help if you start learning the shortcuts, because for multiple repetitive tasks, if you go and use the options in the toolbar, it's going to be a pain. Okay. So if you're in an image and you want to zoom in or zoom out, you can press plus. So when I'm pressing plus, I can zoom in. When I hit minus on my keyboard, I am zooming out. Okay. So that's simple. Then if I want to change my channels, I'm just scrolling my mouse wheel right now, and I'm able to change my channels. All right. If I want to also change my Z with the mouse wheel, I can hit Alter and scroll my mouse wheel, and that lets me change the Z.
Got it. Yeah.
Okay. So that. Sometimes what will happen is the color, the pseudo color of the image will not be saved. So if you want to give a particular pseudo color to the image, you can go to lookup table and say I want this to be green, which it is, but I want to change my red to magenta. So I change that to magenta. Okay. And now it's magenta in color. So that's how you change the colors. So it's lookup table over here, or you can do image, lookup tables, and the same menu also appears over here.
If I have to change
now
in particular color, like I will be opening that particular frame and then change the then go to the lookup table and change it, right?
Right, right.
Mhm.
Okay.
Got it.
Then this menu is really important where you say where you are seeing the image.
Mhm.
It lets you do a lot of things from here.
Mhm.
But before that, something that is very critical is to understand the top part. So C
Mhm.
stands for channel.
Okay,
What channel you are looking right now? First, second, or third channel.
got it.
Z, it's telling you how many Z sections you have in the image and where you are relative to each of them.
Got it. Yeah.
And this tells you the actual size of your image, like 45 microns by 45 microns. And it's a 512 by 512 pixel image. So if I zoom in like crazy, you can see individual pixels over here,
Mhm.
right?
Yeah. Yeah.
So if I start counting, I have 512 by 512 horizontal by vertical. Okay,
Mhm.
so that's that. Um, so few other options that you should always remember is brightness contrast. So you can either go adjust brightness contrast and this menu pops up. You will be using this menu a lot. So
Mhm.
remember the shortcut, which is Shift Control C. Um, and that also pops up when I do Shift Control C. Okay.
Mhm.
And that lets you change the dynamic range.
Got
When
it.
you are changing the dynamic range from here, you are not actually changing actual values. It
Mhm.
is just allowing you uh, to better visualize your um, particular color just because you are
Yeah.
increasing the brightness.
Does it changing just this particular channel or the whole picture?
No, you're only changing this particular channel.
Got it. Yeah.
Okay, so if you see, we have not changed anything over here.
Mhm. Got it. Yeah.
All right, so in this way, you can um, visualize things better if it's very dim. So
Yeah.
this also tells you what is the maximum intensity, in a way, what is the maximum intensity of your um, particular image or a particular object, I should say. Okay,
So
so if we
this can be like these values can be also type of quantification.
no, not really. So we'll get into quantification later
Okay.
when you want to quantify how you do it.
Okay.
But then let's say if you want to play with this image and not actually mess up with your original image, right? So you can right click and do duplicate.
Mhm.
And in duplicate, you can select what you want to duplicate. So let's
Mhm.
say if I want to duplicate just my first channel, right? So I'll enable the duplicate hyperstack. I'll say I only want my first channel, and it's okay if I've got all the slices with me. I'll say okay. So now I'm getting all my slices over here. Okay,
Yeah.
and that's just with this channel.
Mhm.
Now, one thing you will notice is right now my dynamic range is from 31 to 4095, and you can reset this, but somehow Fiji sometimes only lets you uh, do what it wants you to do. But if you want to see from 0 to 4095, you can go to set and say minimum values that I want are 0 and the maximum that I want are 4095. And now you get 0 all the way over here. And what 0 and 4095 really means
Mhm.
is the gray level. So let's go to convert this to gray for a second.
Mhm.
Our eyes cannot differentiate uh, this many levels of gray, which range from a shade of 1 to a
Mhm.
shade of 4095. Our eyes cannot see that. But when you try to quantify, the software can really figure out what shade of gray you are looking at. So if I zoom in to a particular pixel, you can see a different shade of gray, right? Everything in the background, and I want you to focus over here where it says value. So when I'm on a particular pixel, it's showing your value, right?
Yeah,
Although it's black, usually black is going to be 0, and your grays or whites are going to be higher in number. Now, even if it's black, it's never black. There is always a shade of black. So when you have 0, it's black. When you have anything greater than 0, it's not really black, but it's a shade of gray.
There is something which our eyes cannot differentiate between.
Our eyes cannot differentiate. The second thing is there is always some noise in the camera. So either you're looking at noise or some kind of background signal, which is giving you all these values. So let's say if I extend this to, now you can see this other values of grays, right? Which are 57, 65,
so
77.
Even though you change the intensity of the color, it did not change the values, right?
No, it did not. I did not change the intensity. I changed the dynamic range.
Dynamic
right?
brightness.
So and I'll show you what it means to change the intensity real quick. Okay, so this is just changing the dynamic range so that now you can see those lower shades of gray, which you were not able to see earlier. Make sense?
Yeah, it makes sense.
Okay. So your question, which is about changing the intensity, right? So let's say if I crop, duplicate, no. So if I want to crop a small area, I can select this rectangle tool, select this, duplicate, and now it's only going to duplicate this area and nothing else, okay? And let's say if I only want to duplicate the first one. So that's what I got. Well, I did everything, that's okay. So if I zoom into it, right now, the dynamic range is from 34 to 1994, okay? But if you look at the pixel value of the one which is the brightest, it's 1994.
Yes.
Right? Now, if I want to change this permanently, okay? If I want to change it from 1091 to 1994, now what you will see is as I increase my minimum, I am telling the software to ignore everything which is below 595, right? So everything over here, although you read those values right now, which is 52 and whatnot, but when you are seeing it, you are not seeing it. Okay? So let's say, for example, I want to set this permanently, right? So I'll go set and say minimum value displayed is, let's say, 700. So now everything below 700 will be set to 0. And if I do 1500 over here, everything which is above 1500 will be the maximum. So let's say do OK. And what you will see is, and then you apply. Now it gives you a new sort of a value, new sort of a lookup table, right? It's kind of fake, but what it did was everything which was below 700 is now 0, and it stretched everything between 700 to 1600. I guess that was the max, and gives you a new dynamic range value. Okay, that almost falls into the level of image manipulation.
Yeah.
So, but what is not image manipulation is subtracting the background. Let's say if you have a lot of background noise sometimes, it's okay to subtract that. But we can touch on that part at a later stage. Right now, I just want you to teach you basics.
Yeah,
You can practice that for a few days. And once you are comfortable with that, then we can take the next step. Okay?
Yeah.
All right. So this is just playing with the image. Nothing has been changed over here. So I'll bring it to its original values. And that's it. Go to low and so on. Okay. Now, one thing that you will commonly see people do is make maximum intensity projections, something that I have done all the time to see every single particle. So for that, you go to Image, Stacks, Z Project. And you can choose where you want to start and where you want to end. You can select the projection type, whether you want maximum intensity, you want average intensity, you want some of the slices, and whatnot. Okay?
So for our case,
So
we will be doing average intensity.
we will do max intensity.
And why is that?
Because we do max, what we want to do is we want to look at the maximum value for the entire stack. And we know that when you see a maximum value, that's where the particle is.
Okay, yeah.
So if you, let's say if you do average intensity, right? It won't make much of a difference right now. That's because in a single Z plane, when you do Z, it's about voxels and not pixels. Right now, what you're looking at is, yeah, it is a pixel. But when you talk in 3D terms, it's a voxel. In a voxel, depending on the entire, let's say 10 micron height, it will take an average of all of those voxels and present you with the intensity. So if you have multiple particles over there, it will take an average of all those intensities to give you something. Okay? But we are interested in maximum intensity.
But we want maximum intensity of the particles from each plane, right?
Right. So go back to image, text, Z project, and say OK. And it gives me the max intensity of all the particles. And you can really play with that. You know, you just play with these options. It's nothing. I really want you to explore the options that you are seeing. Okay.
So Gaurav, just a quick question. So the image is what I see on your presentation. So that's the maximum intensity of all the stacks you have taken of that time point, right?
Right.
Got it.
I mean, right now I'm just showing you one, not all the time points. So this is,
yeah, no, it makes sense. Yeah, you're just showing something. Yeah, some time point. Yeah, got it.
and this is actually a snapshot. This is not a live time-lapse experiment. Okay. So you end up getting this. That's good. So let's go back to the Z stack again. If you want to look at this in sort of a 3D format, you can go back to image, stacks, and do 3D project. Okay. And enable interpolate, and when you do OK, it will generate a new stack, and you can sort of
rotate it.
Look at that. In a, I mean, this does not have like rich data that you can nicely see things. But let's say if it's a nucleus or something like that, it looks more beautiful. So this looks much better than looking at a grayscale image.
Yes.
So then you can use different lookup tables in which the objects are differentiated based on their location. But we can look at that later. It's not a big deal. So let's just go back to grays over here, and that's what you got. Okay. So now let's go back to the color images. Let's say if you want to merge a couple of colors. And one thing I would tell you is, although I was doing red and green, I would strongly recommend using a combination of green and magenta instead of green and red. That's what I would like to have in the paper instead of green and red, because people who are colorblind, they cannot look at red color. So if we are starting a paper from scratch, not literally scratch, but I'll be happy to redo all the images that I have done and present them as a merge of green and magenta. So when you have green and magenta, the overlay gives you a white color. Okay, so the way to enable the option to get a combiner sort of thing, so go to image, color, and channels tool, right? So this is one more option that you should remember: Shift Control Z. It gives you this channel tool. So let's say if I close and type in Shift Control Z, it opens up. It lets you control different aspects, whether you want to look at a composite image, whether you want to look at a color image, and so on. So the thing is, when you have a composite image, so now it merges all the channels together and displays that to you. You can disable any channel you want to. But one thing is, in this mode, you cannot change the brightness contrast. Okay, it will start changing of the first channel, not the channel that you want to. So if you want to change your brightness contrast, you have to go back to your color mode and then change your brightness contrast. The other way of doing this would be going to adjust and adjust color balance. So when you go to color balance window, even if you are in a composite mode, you can select which channel you want to change. Okay? So there are different ways of changing, and you can always go back to the recording and look at that. So again, this is from image, color, color window. It doesn't show up over here right now because I already have it open here. So let's close this so that you can look at that again. So image, color, no, it's over here, not just image, color, color balance. Okay. All right, so you can change individual channels this way. So clearly over here, this is the particle which is outside because it's both green and magenta. And when we go on to a particular cell, so again, when you are here, there are different ways of panning onto a cell. You can use the hand symbol, and you can move the image you want to, where you want to, if you have zoomed into the image. So let's say if I've zoomed too much, I can move it back and forth with that. And pretty much straightforward, right? So so looking at this, right, everything is magenta, which means all the particles are inside except this one over here, right? So few things. I have saved individual images over here. So if you go back to my analyze folder, you will see that for every image, I have saved individual cell. Okay, and I also have something called cell counter. So let's kind of open this and try to get gist of it. So if I zoom into it, and the way to do this is, again, let's actually go back. You can select individual cell
and then duplicate
and so duplicate that, okay, and you get this.
when you are doing duplicate hyperstake, then it is taking all the channels and planes of that particular cell, right?
That's correct. That's correct. But even before you do this step, I mean, just think about it that we want the maximum intensity projection for all of them, right? So I would rather just do a maximum intensity projection right now. So I'll go to stack, Z project, say okay, we get this, go into one of the images, and duplicate this, say okay, and we got this, right? Now we can save this the way I have saved over here, right? So make sure you create.
But Gaurav, for this particular image where you took the maximum intensity projection and then took one cell out, is it I'm just thinking if we take one cell out and then do maximum intensity projection, it is going to do show the same thing, right?
Yeah, it will show the same thing.
Okay, got it. Yeah.
Let's say we zoom into it. So one thing you will see is when you do maximum intensity projection of transmitted light image, it does not look good. And I'll pull up a plugin which lets you do this properly. Or actually, it shows you a good image. I'll share that plugin with you later. It's not something which is critical right now. But these are the two channels which are important, right? So we come back over here and we can, you can either manually count it, right? Because there aren't many particles. But what if there are plenty of objects that you are seeing over here? How do you handle something that complex? So for something like that, you can, so let me convert this. Actually, I was going to save this. Sorry. I went way ahead of what I wanted to do. So I'll go and save this. So make sure you create a lot of folders like this. Go to File, Save As, TIFF. Okay?
Yeah.
And make an appropriate folder and save it over there, and that should be it. All right? Few other things. So let's go back over here. What is very important is to have the right scales. And the scale analyze set scale. So by default, when you go and analyze set scale, it should be able to use the data from the metadata of the image. So it's telling you right now 11.3 pixels equals to 1 micron.
Okay.
Okay. So we can do math to understand what is the size of the particle that we are seeing. All right. So let's say if we do cancel and we want to measure the size of the particle, what you can do is uh, just go and click on this tool and draw a line across that, okay? And when you are drawing a line across, it's telling you land on something over here. So if you just focus on this area, when I draw the line, it tells me what's the length. And that's about 67 microns. 670.68 micrometers, sorry. So it's 0.600 microns or 700 microns, okay? So it's about whatever nanometer that turns out to be. And if we kind of go from end to end, so it says 45 microns by 45 microns. So if I start doing this, now it's 45 microns by 45 microns, right? So if we do this, this is about 0.6 microns or 690 nanometers. Make sense?
Yeah.
Okay. So with that, let's go back over here. Let's crop this guy. And let's just do 1 and 2. So let's say if I'm in my first channel, let's go to color mode. That's way too bright. So I want to try and count this guys automatically. Okay?
Yeah.
So what I'll do is I'll try to draw a mask on it. So what I did, I tried to threshold. So image adjust threshold. That's Control Shift T. Okay, that's what I press the shortcut. Control Shift and T. Now you can set a mask to count individual particles. So I'm setting up a mask over here, but you will see that there are some particles which appear as one. For example, that's over here. Let's say I apply this mask on them. So now it created a new masked image. Image process. Go to binary, convert to mask. It's a mask now. And then if you want to separate some of this closely associated pixels, you can try out different options. So go to binary. You can dilate them. No, that's the wrong one. Process, binary, erode. No, it's also a wrong one. Actually, watershed, sorry. So the watershed did not work out because things are way too close over here. Let me try again and see if that works. No, so it's not working out. So that's one of the limitations that sometimes
Gaurav, after you selecting watershed, you are clicking no.
Because I only want to process one channel, not both the channels.
Okay.
So process
Got it.
um, binary, watershed.
Asking,
So
oh,
do you want to do both? I said
got it.
no, but yeah, so it doesn't change anything, right? So it's
Yeah.
fine. So and then you can go to plugins, no, analyze particles, and you can measure, you can select the size of the particle you want to measure, and so on. So let's say if I'm doing 0 pixels, let's just do pixel units and not size. Okay, so now we can say how many pixels I want in particles. So it goes from 0 to infinity. Uh, and these are different things you can play with. So if I do OK, say no, it should give me,
It's 27K is the number of pixels.
no, it should, where did that go? Hold on. So don't do anything. Hold on. Analyze, analyze particles. Oh, display results. That's why. Okay. Display results, add to manager, summarize. Okay. So now it tells me how many particles I have, right? What is the area that it's occupying, and so on. So this is the image now. Of course, this is not right? These are two particles and not one particle. So that's something that you have to manually adjust for. The way I have created my data is depending on the concentration of the particles. Uh, I have what I have mentioned is, and I did not go with counting number of particles. I went with the area that the particles are occupying, right? So that's another thing, another way of doing. So we can figure out how we want to do this, whether we want to do this based on number of particles, count it that way, or you want to count, do the area wise. So that's something which I'll clear up my mind on later on. But this is one way that you will do, right? So you can create masks, figure out number of particles, write that, maintain an Excel spreadsheet, and so on. So that's that. And the other thing is in this
So
ROI manager.
can I copy? So I will just copy the count, or can I copy this whole? Can I also save this file where it
Yeah,
has?
you can. So what I like to do is I like to save this ROI manager. Okay. What ROI manager in future, if somebody tells you, oh, show me what you counted, you can. So let's do this. Let's do this. Okay. More saved. So let me just go back and folder. All right, so I saved this ROI file over here, the ROI manager, right? It's a terrible name. Of course, you want to have a nicer name than this. So let's call it Sal1. And what I'll also do is I'll save this guy. Okay, so let's say Um, so let's save this. Call it max projection cell 1 max projection. That's it, and this is the mask, right? So I'll save this as cell 1 mask. And let's close everything so that we can duplicate all of this. So I have my cell 1 max projection image, drag it, I get this, all right? Now if I want to know individual particles over here, I can open up my cell 1. Oh. I'll do this later. So what you do is you hit T. So when you hit T, it opens the ROI manager. Okay. And
so which
from
one did you press? The T. Okay, got it.
yeah, T as in Tom.
Mhm.
So you press T, and then you go more and say open, and I'll open this ROI manager, right? So I'm so glad I'm doing this. I forgot this. I'm doing this after a long time.
No, no.
Okay, let me do this again. It only selected one ROI and not everything, and I'll tell you why. So let's just delete this, and that's totally my fault because I totally forgot about this. This is what happens when you do something after a long time.
Yeah, no.
So let's duplicate this guy again. Actually, I can. No, that's okay. Uh, Surprisingly, everything is labeled. I don't want it that way. So duplicate. To go back to the threshold, select the right threshold. So clearly there are two problems happening. These are two particles, these are two particles, but with the way I'm thresholding, they will be counted as one particle, right? So that's something which you have to adjust by yourself, and we can work on this later to get things better. Apply. So that's a mask now. Analyze particles. We want all of this, say okay. And we get the ROI. All right, so what you have to do, in fact, is not just save it right away, but you have to select all of them. So you, let me go back. You click Shift and select all, okay? And then you go and do save. If you don't do it that way, only the first ROI gets saved.
Okay.
All right, so let's close all these guys. Let's duplicate this again, open up this. And now if I press C, C as in Tom, go to open, and now you can see it's a zip file kind of thing, right? Open the zip file, it shows you all the ROIs.
Got
and you
it.
can click on each of them, and clearly it shows you that this ROI has got two particles, but of course it's getting counted as one,
Got it.
right? So now you can go through every single particle and you can figure out how many particles you have. So, I mean, this is, you can count manually, but the thing is, we can do something called batch processing where we have got all these files. We can create a small macro in FIJI, which lets you do all these things automatically.
Okay.
Okay.
Gaurav, just a quick question. So when we are so for the uptake experiments, we'll be counting the particles, right? Not the intensity.
We can do it both ways. So let me go back and look at my presentation, how I've done. And I don't think I counted intensity. What I counted was area.
Mm-hmm.
And I think I did count intensity because if more particles are taken up, the intensity will be higher at the same time.
Yeah. Yeah.
But now the thing is, all of this depends on the image settings. So, the settings that I use versus the ones that you will use will be different.
Mm-hmm.
Uh, so intensity probably will not be the right way unless we normalize it in a some way. That's a different story. But if we don't normalize it, it might be a problem. But I think what will stay consistent always is the area
Mm-hmm.
and the number of particles,
Yeah, got it.
right?
Mm-hmm.
So we can try to aim to get those two metrics.
Yeah, yeah, I think that would be like more, I mean, clearer thing to do, like more simple thing to do.
All right, so that's something that I wanted to show,
Mm-hmm.
relevant to everything that you will be doing.
Mm-hmm.
There is a lot of stuff that you can do,
Mm-hmm.
and as in when I sit down and do more of the analysis on this, try to create macros and whatnot, and we can get into macros sometime later. For
Mm-hmm.
the moment, just practice all of this.
Yeah,
Uh,
I think these are completely new to me, so I'll practice them and also I'll go back to the video. So yeah, so that I am get used to with it.